All right, so our next lecture in talking about the metabolism of glucose, here we're going to talk about the synthesis of glucose within the cell, which is called gluconeogenesis. So first, just an overview of gluconeogenesis. It's the synthesis of glucose, like I said, from non-hexose precursors. So that's a really important distinction. And so you'll also hear this referred to as de novo synthesis. So forming glucose from glycogen, like we talked about in the previous lectures, glycogen breakdown, forming it from fructose or galactose, that is not gluconeogenesis. So gluconeogenesis is forming glucose from essentially non-carbohydrate precursors. And you'll see where that comes into play here in a second. So the precursors of gluconeogenesis are pyruvate, lactate, amino acids, except for leucine and lysine, propionic acid, no other fatty acids are involved, and then glycerol, which also comes from triglyceride breakdown. So gluconeogenesis occurs during prolonged fasting and starvation, and then it also occurs after meals that don't contain carbohydrates. So Atkins diet or similar types of diets where the person has very limited or no carbohydrate consumption, that can also trigger gluconeogenesis. And so essentially you want, again, with these biochemical pathways, you want to be thinking about in response to mainly the fed state or the fasting state. And the starvation state's just a prolonged form of fasting. And so you want to think about this as a response to fasting or starvation, and it's a way to provide glucose when there's a very low level of glucose in the body. And so the main site of gluconeogenesis is the liver, as we've talked about many times before in previous lectures. It can also occur in the kidney, but this is really in response to kind of acid-base situations and amino acid metabolism. And so we'll talk about that in a little more detail in the amino acid lectures. So it's mainly occurring in the liver. And remember, the reason it's happening in the liver is the liver during fasting relies on fatty acid oxidation, which allows it to turn off glycolysis and turn on gluconeogenesis. Because you remember, you can't have both running at the same time. That would be counterintuitive. And so if you remember, you're trying, the name of the game in the liver during fasting is free glucose that can then be exported out into the bloodstream and used by other tissues. And the two main sources of that are glycogen, so glycogen breakdown, and then gluconeogenesis. And remember, you have to use gluconeogenesis because eventually you're going to run out of glycogen. So this is a figure, it's in your book. We're going to go through this figure entirely during this lecture. And just to kind of give you a brief overview of the figure here, because could be kind of confusing when you first look at it. We tried to simplify it as much as possible. So the main starting point here is pyruvate. Now you can get to pyruvate from alanine, which comes from amino acid metabolism. You can get there from lactate as well. And then you come down here in the mitochondria where pyruvate in the first committed step is converted to oxaloacetate by the enzyme pyruvate carboxylase. So we have a nice legend here for some of these key enzymes where we had to abbreviate them. And then you'll notice where when you get to oxaloacetate, it's kind of a fork in the road. You can go towards phosphoenol pyruvate, or you can go towards malate, which then gets exported out and then converted back to oxaloacetate. And we'll talk about in more detail why that happens, but the main driver of that is actually the levels of NADH in the cell. It's kind of where your starting point is, whether it's pyruvate or lactate or alanine. The other thing we'll point out here is glycerol can also be used in gluconeogenesis, and that comes from the breakdown of triglycerides in adipose tissue. And then the last thing here we'll point out is you may notice if you compare this to a diagram of glycolysis, there's a few steps at this point here that we leave out because they're not very significant steps. They're not major points of regulation or anything like that. We'll certainly mention them later in the lecture, but we left them out of the figure just to keep it more simplified. Once we get up here, you'll notice it's just kind of going up in a reverse fashion through the steps of glycolysis, with the exception of a couple enzymes, and then you get to your final point here of glucose. And again, the other thing I'll point out here is blue is where it's only happening in glycolysis. So you can see that here where it's unidirectional. And then gluconeogenesis is where you have enzymes that are specific for gluconeogenesis. So glucose 6-phosphatase, remember we've talked about this, fructose 1,6 bisphosphatase, it has a green arrow. Bidirectional means that it can happen in glycolysis going down this way or in gluconeogenesis coming back this way. All right, so now we're going to go through the steps of gluconeogenesis and talk about the regulation along the way. So the starting point again is pyruvate. There's a couple ways you can get there. So first with alanine. Alanine, remember, comes from protein breakdown in the muscle. And remember, we talked a little bit about this in the glycolysis lecture. So that comes into the liver. So you have alanine here. And then alanine undergoes a process called transamination, which uses alpha-ketoglutarate and combines with alanine, and then produces pyruvate and glutamate. And this is a very common process in amino acid metabolism, and we'll talk about this in much more detail in those lectures. But the bottom line is you can get alanine, use this alpha-ketoglutarate, and you produce pyruvate. And then this is catalyzed by an enzyme called alanine transaminase. On the other side here, you have lactate. So if you have a buildup of lactate, this can then be converted into pyruvate via the enzyme lactate dehydrogenase, as we've indicated here in LD. We have it here. And again, this is also influenced by the levels of NAD+ and NADH. So if you have high levels of NAD+, that also favors pushing this reaction towards this way. Because remember, this is a reversible enzyme. During anaerobic respiration, when you're carrying out glycolysis, the last step here is you convert pyruvate into lactate. During gluconeogenesis, it carries out the opposite reaction. And then what's also important to note is that it produces an NADH during gluconeogenesis, which then can be used up here by the glycolysis enzyme glyceraldehyde 3 phosphate dehydrogenase. Except in this instance, it's going to use it in its gluconeogenic function of converting 1,3-phosphoglycerate into glyceraldehyde 3 phosphate, which then produces a NAD+, as we show here. So if you remember, during glycolysis, phosphoenol pyruvate is converted into pyruvate by the enzyme Pyruvate Kinase, PK. This is an irreversible enzyme, and as a result of that, during gluconeogenesis, when you have pyruvate, you can't just convert it back to phosphoenol pyruvate using this enzyme because it's irreversible. So you have to kind of find a more roundabout way. So there's some alternate pathways that pyruvate undergoes. So pyruvate goes into the mitochondria, and as you remember, during aerobic respiration, it can be converted into acetyl-CoA, as we indicate in the blue arrows here. Remember, the blue arrows indicate glycolysis, and then it can go into the citric acid cycle and so on. However, in gluconeogenesis, it's going to undergo conversion into oxaloacetate by the enzyme pyruvate carboxylase, as we've indicated here in PC, if you look here. Now, this is the first committed step of gluconeogenesis from pyruvate. And remember, when we're talking about metabolic pathways, that's really important. The committed step, the rate-limiting step, steps of high levels of regulation, all important things to pay attention to. Those are where test questions come from. So one thing you'll note here is if you have a buildup of acetyl-CoA on the regulation side of pyruvate carboxylase, acetyl-CoA actually stimulates pyruvate carboxylase. And that's important because remember, in the liver during fasting, we've talked about this, the liver switches from hepatocytes, the actual cells of the liver switch from using glycolysis as their major source of energy to using fatty acid oxidation. So they break down fatty acids for energy. And remember, the reason they do that is so they can carry out gluconeogenesis. So you can see really where it's kind of cool. This is all tying together. So you break down fatty acids. Breaking down fatty acids produces a lot of acetyl-CoA, which helps gluconeogenesis here. And this is the process of beta oxidation. So beta oxidation is the breakdown of fatty acids to acetyl-CoA. The other thing that beta oxidation produces is NADH and ATP, obviously. And so this also helps with gluconeogenesis because, as we say here, it helps provide the energy for gluconeogenesis because NADH is a major factor used in gluconeogenesis, and then obviously ATP provides energy for it. And then again, by producing the end product of acetyl-CoA, that helps stimulate the first committed step here. All right, so as I mentioned when I was giving an overview of this figure, when you get to oxaloacetate, it can kind of take one or two routes. So it can go directly to phosphoenol pyruvate, or it can get converted into malate, which is then exported out of the cell. So first we'll talk about the route number one. So oxaloacetate, it gets converted into phosphoenol pyruvate by an enzyme called phosphoenol pyruvate carboxylase, or PepsiK. We also indicate that in here. And so this is the mitochondrial form of PepsiK. There's also a cytosolic form, which we'll talk about on the next slide. It uses up one equivalent of GTP, as we say here. And what's important on the regulation side is that glucagon stimulates the synthesis and activity of PepsiK. And that makes sense. Glucagon is synthesized and secreted during the fasting state. Gluconeogenesis is occurring during the fasting state. And the other thing is, glucagon stimulates the synthesis of a lot of different gluconeogenic enzymes as well. Because you're in the fasting state, you want to synthesize glucose. That makes sense that glucagon is going to stimulate gluconeogenic enzymes. The other thing is oxaloacetate itself stimulates PepsiK as well. And that makes sense. If you have a buildup of oxaloacetate, and you're in the fasting state, it's going to help stimulate this enzyme to help carry this reaction forward. So this was route number 1, where in the mitochondria you go directly to phosphoenol pyruvate. That gets exported out into the cytosol, where it can then undergo the rest of gluconeogenesis. Route number 2, though, involves converting oxaloacetate into malate via malate dehydrogenase. This uses up an equivalent of NADH. And the reason you convert it into malate is because oxaloacetate itself cannot be exported out of the mitochondria, but malate can. So you convert it into malate, malate gets exported out into the cytosol, and the cytosolic form of malate dehydrogenase converts malate into oxaloacetate, so converts it back, and then it also gives you back an NADH, so it produces an NADH here. Oxaloacetate is converted into phosphoenol pyruvate in the cytosol by the cytosolic form of Pepsi-K, and this as well uses up an equivalent of GTP. What's important to note here is this NADH here, generated when you're converting malate back to oxaloacetate, this NADH can then be used by glyceraldehyde 3-phosphate dehydrogenase. Now you may be wondering, what forces oxaloacetate to take route number 1 versus route number 2? And that's a really important question. So it all stems back to how you made pyruvate. So when pyruvate is formed from lactate, so we start back over here with lactate. Remember, lactate gets converted to pyruvate via lactate dehydrogenase here and produces an NADH that can then go on to be used by glyceraldehyde 3-phosphate dehydrogenase in this conversion of 1,3-bisphoglycerate into glyceraldehyde 3-phosphate. And so when you use lactate as your starting point, oxaloacetate can be converted directly into phosphoenol pyruvate within the mitochondria by Pepsi-K. On the flip side, when you use alanine, you don't generate any NADH like you do when you use lactate. So alanine gets converted into pyruvate, comes down here in the mitochondria, gets converted into oxaloacetate, and instead of going down route number 1, it's going to go down route number 2 to form malate. Malate gets exported out, gets converted back to oxaloacetate, which also generates an NADH in the process, and then oxaloacetate obviously is converted into phosphoenol pyruvate by Pepsi-K, and this NADH that was produced by malate dehydrogenase is then used by glyceraldehyde 3-phosphate dehydrogenase. And so as you can see here, it matters where your starting product is. If you start with alanine and you don't produce any NADH, an equivalent of NADH to be used by glyceraldehyde 3-phosphate dehydrogenase, that's going to force us to go route number 2 so that you can still get that NADH here. Versus if you start with lactate, you've already produced an NADH when you convert lactate into pyruvate, so there's no sense in going down this other pathway here. You can just go down pathway number 1 and directly convert oxaloacetate into phosphoenol pyruvate in the mitochondria, which then gets exported out and then can undergo gluconeogenesis in the cytosol. So you can see, regardless of which route you take, you end up with the same end product, which then undergoes gluconeogenesis. And so as you can see, it depends on what your starting point is. And then there's also, in addition to that, the balance of NADH and NAD+ in the cell, which you can see that balance influences a lot of different metabolic processes, glycolysis, gluconeogenesis, and so on. All right, so again, regardless of whether you took route 1 or route 2, you've ended up here at phosphoenol pyruvate. And what's important is you've gone through all this work to get to phosphoenol pyruvate because pyruvate kinase is an irreversible enzyme that converted phosphoenol pyruvate into pyruvate during glycolysis. And the last thing you'd want to do is go through all that work and then have it again be converted back to pyruvate. That would be a futile cycle. So during gluconeogenesis, pyruvate kinase is actually inhibited for that specific reason. And it's inhibited by a number of things. It's inhibited by alanine. So you have increased levels of alanine, which can come back here, inhibit. And remember, that's coming from muscle tissue, protein breakdown during fasting. Also increased levels of ATP. And remember, that's coming from fatty acid oxidation. So fatty acid oxidation, you get acetyl-CoA. And then you also get ATP, which helps inhibit pyruvate kinase as well. And then also fructose 1,6-bisphosphate. This is an intermediate during glycolysis, and since you're not running glycolysis, you have less levels of that, which during glycolysis, this is actually a stimulant of pyruvate kinase. So you have less levels of that as well. The other thing in the liver, in hepatic pyruvate kinase specifically, is inhibited by phosphorylation by protein kinase A. And remember, during fasting, you have increased levels of glucagon, which then stimulates cyclic AMP, which then gives you protein kinase A. And then protein kinase A inhibits pyruvate kinase as well. And so there's, in the liver, which is the main site of gluconeogenesis, glucagon and then subsequently protein kinase A are also going to play important roles as well. All right, so now that you have phosphoenolpyruvate and you're inhibiting pyruvate kinase so that it cannot go back to pyruvate, the next logical step is for it to go up through the steps of gluconeogenesis to result in free glucose, which can then be used by other tissues. And as you can see in the diagram here, from phosphoenolpyruvate to glyceraldehyde 3-phosphate, there's actually a number of steps between these two intermediates here that we cut out because they're just not too important. And we'll just run through them very briefly on the next slide so you're aware of them. You'll see we took this from our glycolysis diagram because all of these enzymes, these glycolytic enzymes, they're reversible enzymes and they can run in reverse during gluconeogenesis. So here we start with phosphoenolpyruvate, and this gets converted into 2-phosphoglycerate by the enzyme enolase. Then that gets converted into 3-phosphoglycerate by phosphoglycerate mutase, and then 3-phosphoglycerate gets converted into 1,3-diphosphoglycerate using a phosphate from an ATP. So again, another example of an input of energy. And then lastly here, this is an important step. This is included in the other diagram as well. 1,3-diphosphoglycerate gets converted into glyceraldehyde 3-phosphate using up an NADH. Remember that NADH is really important. We talked about in the previous slides. And this is catalyzed by the enzyme glyceraldehyde 3-phosphate dehydrogenase. All right, so now that you've formed glyceraldehyde 3-phosphate, one thing before we go to the next step, there's really two ways you can get glyceraldehyde 3-phosphate. There's the way we just talked about where 1,3-bisphosphoglycerate is converted to it by glyceraldehyde 3-phosphate dehydrogenase using up that NADH. The other way involves fatty acid metabolism. So if you remember, during fasting, the liver uses fatty acids instead of glycolysis to produce ATP. So what that involves is breaking down triglycerides in adipose tissue into fatty acids and glycerol. And so the fatty acids, obviously, they will undergo beta oxidation to give you acetyl-CoA. And then the glycerol that's broken down in that process can also contribute to gluconeogenesis. So it enters here, and then it's converted to 3-glycerophosphate, which is then converted into dihydroxyacetone phosphate, which, if you remember from our glycolysis lecture, is actually rapidly converted to glyceraldehyde 3-phosphate. And so that's another way that you can get to glyceraldehyde 3-phosphate in a way that fatty acid metabolism also contributes to gluconeogenesis. Again, you can see where all these processes are tied together. Now that you have glyceraldehyde 3-phosphate, that gets converted into fructose 1,6-bisphosphate by the enzyme fructose 1,6-bisphosphoaldolase. All right, so now that you've formed fructose 1,6-bisphosphate, the next step is then to convert it into fructose 6-phosphate. This is catalyzed by an enzyme specific to gluconeogenesis, which we've indicated here with the green arrow, and that's called fructose 1,6-bisphosphatase. So it's essentially taking the phosphate group off of this one position to yield fructose 6-phosphate. I've also modified the figure here a little bit on this slide to indicate that this is also, if you're going down this way from glucose through glycolysis, phosphofructokinase, or PFK1, catalyzes this step here in blue, the conversion of fructose 6-phosphate to fructose 1,6-bisphosphate, which, if you remember, is a committed step, meaning it's irreversible. So that's why you need this gluconeogenic enzyme. It's irreversible, and it's the committed step, and it's very highly regulated. Now, so back to fructose 1,6-bisphosphatase, the levels of regulation for that enzyme, you have ATP and glucagon are positive regulators. So you've got ATP here, which is coming from high levels of fatty acid oxidation, and that's a positive regulator. And remember, you have high levels of fatty acid oxidation because in the liver, you've switched from using glycolysis to using fatty acid oxidation to provide ATP so that you can carry out gluconeogenesis. And then remember, during the fasting state, glucagon is released, and glucagon contributes toward the synthesis of fructose 1,6-bisphosphatase. Now, on the flip side, remember when you're going through glycolysis, fructose 6-phosphate, some of that can be shunted over here and converted into fructose 2,6-bisphosphate by the enzyme PFK2. And remember, fructose 2,6-bisphosphate is a positive regulator of PFK1. Now remember, you want to think of it as kind of an on-off switch in a sense. When you have glycolysis on, you want to turn off gluconeogenesis. When gluconeogenesis is on, you want to turn it off. So fructose 2,6-bisphosphate kind of dually contributes to that. It helps turn glycolysis on by stimulating PFK1, and then on the flip side, comes over here and inhibits fructose 1,6-bisphosphatase, which also contributes towards running glycolysis. The other thing is AMP is also a negative regulator of fructose 1,6-bisphosphatase, and remember AMP is a positive regulator of PFK1, so same concept. And remember, AMP reflects the energy state of the cell. In low energy states, you're going to have high levels of AMP, hence why you're carrying out glycolysis, because you want to eventually produce ATP to provide energy for the cell. So again, AMP levels influence these two enzymes as well. Now again, remember fructose 2,6-bisphosphate is a positive allosteric regulator in glycolysis. And then remember, it's a bidirectional enzyme. It has kinase activity, which converts fructose 6-phosphate to fructose 2,6-bisphosphate. And it essentially adds a phosphate group here to the 2 position. And then remember, PFK2 also has phosphatase activity, which converts fructose 2,6-bisphosphate back to fructose 6-phosphate. And during the fasting state, you have high levels of glucagon. Glucagon, remember, stimulates cyclic AMP, which then stimulates protein kinase A. And then protein kinase A actually stimulates the phosphatase activity of PFK2. And by doing that, it's going to stimulate the conversion of fructose 2,6-bisphosphate to fructose 6-phosphate, which decreases this, and then removes fructose 2,6-bisphosphatate's ability to stimulate PFK1, in essence, turning PFK1 off. And then in addition to that, since you have decreased levels of this, it also can't contribute to inhibition of fructose 1,6-bisphosphatase. And so what the end result is you have decreased glycolysis and increased glucosynthesis or gluconeogenesis. And so you have stimulation of this enzyme. All right, so now you've formed fructose 6-phosphate. That gets converted into glucose 6-phosphate, which is that key intermediate we've talked about in previous lectures. And the enzyme that carries that out is phosphohexose isomerase, which by the bidirectional arrow here indicates that it's an enzyme that's reversible, involved in glycolysis, and then for these purposes in gluconeogenesis as well. So now that you get to glucose 6-phosphate, remember, when glucose comes into the cell, it gets immediately phosphorylated by either glucokinase or hexokinase to prevent glucose from leaving the cell, because remember that phosphate group prevents it from crossing the cell membrane. So in this last step here, you got to take this phosphate group off of this 6 position. And so that's where glucose 6-phosphatase comes in. And that's a gluconeogenic enzyme, which is indicated by this green arrow here. And remember, this is the enzyme that's deficient in von Gurke's disease. And so this carries out that last step, gives you free glucose, which then can be used by other tissues, which is the whole point of gluconeogenesis. Now on the regulation side, remember during the fasting state, you have glucagon, which stimulates the synthesis of glucose 6-phosphatase. So glucagon stimulates it. And then remember in the fed state, you have insulin. And so you don't need to, in the fed state, you have plenty of glucose around. You don't need to synthesize more of it. So insulin's actually going to decrease the synthesis of glucose 6-phosphatase. And then lastly, glucose 6-phosphatase is actually stimulated by glucose 6-phosphate. So if you have a buildup of this, you're in the fasting state, it's going to help drive this activity of this enzyme to help drive towards producing free glucose. All right, so lastly here, this is just kind of a summary slide that ties together a few points I've made throughout the lecture. So during prolonged starvation, glucagon is elevated, which we've mentioned, and it stimulates the synthesis of gluconeogenic enzymes. Specifically, it stimulates the synthesis of PepsiK, both the mitochondrial form and the cytosolic form. It stimulates the synthesis of fructose 1,6-bisphosphatase, and then glucose 6-phosphatase as well. So these 3 enzymes are the main ones it stimulates the synthesis of. At the same time, it actually decreases the concentration of glycolytic enzymes, such as pyruvate kinase, PFK1, and glucokinase. All right, so that closes out our discussion of gluconeogenesis. Thank you for watching this sample lecture from our online biochemistry video course. You can access the entire video course and the corresponding outline format textbook on our website using the link in the description below.